Okay, good morning, everybody. Welcome to another one of our Thursday seminars, Research Horizon seminars. To this Thursday, we have two speakers. The first one is Ilayda Bozan, and Ilayda comes from the Strombeck Lab. Ilayda obtained her bachelor's in bioengineering from Marmara University in Istanbul, Turkey, with honors degree in 2018. She studied the effects of amino acid interactions to human leukocyte antigen global dynamics. And now she's studying structural variation in landrace potatoes through de novo genome assembly and genome comparison. Um, just to remind everybody, since we have two speakers today at the end of each seminar, please don't forget to fill out the survey. Okay, go ahead, Ilayda.
Hi everyone. Today I'd like to talk to you guys about our project on genome analysis of the diploid wild potato species Solanum bukasovii. First, oh, it's fast. First, I'd like to talk to you about what is Solanum bukasovii and what are available reference genomes and what methods and we used and what results we got from them. First of all, potato is the economically most important vegetable crop in Canada, and it is the fourth most important crop in the world. We got this image from International Potato Center in Lima, Peru, which their gene bank has over 4,500 native species of potato. Solanaceae is a very important family, which includes tomato, pepper, aubergine, and tobacco, alongside with potato. The chromosome number for potato is basic chromosome number is 12, and the ploidity in potato ranges between diploid to hexaploid, but most of them are tetraploid, and the heterozygosity in potato is very high. That's why what we think a real potato is this. There's all sorts of potatoes looking very differently. A reference genome is a digital nucleic acid sequence, and it can be used for structural variation analysis and help genome assembly. But some problems with the reference genomes are that it doesn't always represent the heterozygosity in the taxon, and most of them are incomplete. A genome assembly can be done in three ways, which are reference genome guided assembly. The good thing is it is easy, but the problem is that there aren't many options when it comes to a reference genome. Your species might not have a reference genome available, and it will bias the assembly towards the reference genome that is used. De novo assembly is to using short reads or long reads or both of them together to overlap and assemble it together. Mostly used when a reference genome is not available, and it'll create independent results. But the problem is it is difficult and it takes long time. A hybrid approach is to use both of them together when again a reference genome is available, and it can be it can help the assembly, but at the same time, it will bias the assembly towards the reference genome that is used. And structural variation in the genome is the copy number variations are where the specific regions of DNA are copied or deleted. These may cause the gene expression levels to be increased if it's doubled. It could decrease when it's deleted. And it's been shown that CNVs in potato diploid species affect pathogen resistance and antibiotic tolerance. So there are 6 reference genomes available for potato. They are all different species. First of all, DM is a doubled monoploid, so it is not real. It's just a doubled monoploid clone of Solanum tuberosum group Puerha. And RH is a very new reference genome. Now it's available. It is tuberosum and it's the parent of DM. M6 is very important because it has many economically important traits. Gonia calyx is a diploid cultivated potato species available from our lab. Solentus is a commercially available potato. It is from Solanum tuberosum, and Solanum commersoni is a very fragmented assembly, so it's kind of a unusable reference genome. Solanum bukasovii is a diploid wild species, and it is very important because it bridges the wild potatoes to the cultivated potatoes. It closely related to the cultivated potatoes. And in our lab, it's already been studied with illuminarids, and to further investigate, it was resequenced with 10x genomics, and previous findings from our lab shows that bukasovii 1 and bukasovii 2 chloroplast has the highest SNPs compared to each other within a 12 other potato species. When we're exploring the genome of Solanum bukasovii, I have two hypotheses. First of them is that when compared bukasovii 1 and 2 have SNPs compared to each other, and bukasovii 2 has structural variation compared to other reference genomes. In order to achieve these, we have objectives of first de novo assemble, get the reads, de novo assemble the bukasovii 2, then use the available sources to do a structural variation analysis against bukasovii 1 and the other available reference genomes, and then automate this pipeline in our portal that we create and visualize our results. First step is to de novo assemble the Solanum bukasovii. Since we had 10x genomics link reads, we used Supernova and Long Ranger, which is available through 10x. And in order to de novo assemble it, after we use mummer and bed tools to remove any contaminants, and mitogenome and plastome with the help of Sai from our lab. And then we use Pigment and arcs to further scaffold the assembly we had. And our results show that we have now 11,800 contigs, our assembly, and it is 617 megabase long. And Busco analysis show that it has 5,761 complete Buscos, which is 94% complete Buscos. And the second analysis was the structural variation analysis of Solanum bukasovii. For this, the methods we use is we use BWA for alignment, SAM tools for alignment manipulation. We used freebase and SNPhef to call SNPs, and Cyaminator to call CNV. But the problem with that is that we didn't get any results from this, and we had to take a step back and re-evaluate our pipeline. And we found another tool that actually takes advantage of the 10x genomic reads barcodes that in order to align the reads that we have to the reference genomes. So we redid the pipeline and used the barcode aware reference genome aligner and did the same steps. And at the end, we were able to get some results. In this page, we can see the results, and it shows that definitely when compared BWA to the Long Ranger pipeline has better results. It aligns better and it aligns to a deeper level. And when we look at what these are telling to us, we see that DM and Solentus has aligned a lot, and bukasovii 2 obviously comes very second because it is very close to bukasovii 1. When we look at the SNP results, Solentus has the highest SNPs compared to bukasovii 2, and intergenic SNPs are higher than the exon or intron SNPs. And when we look at the CNV results, The Solintus again has the highest number of CMVs compared to bukasovii 2, and then DM and Gonya calyx 1. When we look at the CMV results, it's shown that most of the time, development proteins, stress response proteins, detoxification proteins are the ones that are mostly affected. And for the last part of the project, we developed the potato genome portal. And this is the potato genome portal that we created. We did this in order to show our results. Right now we have 13 of our genomes and their chloroplasts, and very soon we will have their mitogenomes available as well. And in this, we wanted to automate and be able to uh, help our collaborators work on the projects with us as well. So we created a data sharing platform between us so they can upload their data and we can automate this project that we are going to do in the future. And if we look at what we found from bukasovii 2 is that we checked both of the objectives and both of the hypotheses, I'm sorry. And there are SNPs when compared, bukasovii 1 and bukasovii 2 are compared. And there's structural variation between bukasovii 2 and the other reference genomes available. And all the objectives are met. The bukasovii 2 was assembled. Right now it is assembled into 11,800 contigs. There are SNPs, there are CNVs, and the pipeline has been fully automated, and we can visualize our results using the portal. And these are the references. And I'd like to thank everyone who's been involved in my studies. I think without any one of them, I wouldn't be able to come where I am right now. And I'd like to thank everyone for listening. And if you guys have any questions, I would love to answer them.
Okay, thank you, Ilayda. Okay, so we have quite a bit of time for questions. So please you can post them on the chat, unmute yourself, and ask the question. Or for those of you listening on YouTube, you can just write them on the chat, and I will ask the question.
I, I can ask a question.
Of
I have a
course.
question. Nice talk. I guess my question would be, when it comes to potato breeding, because I'm sure your tool that you're developing is really to be used in a way, for potato breeding. So what would be the biggest challenge right now when it comes to potato breeding?
I kind of didn't work on the breeding side. I was more involved in the investigating the genome side, but of course, all of our studies are to help the breeders in the end to have potato species that are disease resistant and economically important, right? So how will this help to the breeders is that we will be able to see where exactly in the genome, when it's compared with the other ones, is the trait that we're looking at. For example, if we're looking at a SNP, where does it lie? Or if we're looking at a deletion, if it is compared with one genome, it is there, and when it's compared with the other genome, it's where is it? So we will be able to visualize all of these.
Okay, thank you.
Thank you.
So you have the doubled monoploid reference.
Yes.
And now that's your main reference, right?
Yeah, we have 6 main references, but if we actually look into it, 3 of them are the main references, and RH and Solintus are very new. They were published a month or two ago, so we're just tapping into them actually.
Okay.
And that was the problem with the DM. DM is doubled monoploid. So even though it is a reference genome that is created to decrease the heterozygosity in the potato and to be able to create a reference genome, it still does not help because it is not real.
Yeah. So with new structural variation characterization, then say next time you go to genotype of population um, of Solanum, how is this other species going to, I guess, what's going to be the relationship when you try to transition more towards the towards tuberosum rather than?
So potato is very big, and there's uh, a lot of debates on how to divide the species. The people are not agreed in one way. There's a lot of voices, and I think we cannot say that each species will be able to, like I don't think we'll be able to do that. But I don't know if I'm answering your question.
No, that's understandable. Thank you. Just wait a few seconds for any other burning questions. Okay, well, if there are no more questions, thank you, Ilayda,
Thank you
and
for your question.
we can move on. Thank you. Okay, you can stop share, and then Angela can share her presentation.
When I figure it out where my Zoom is, I'm sorry.
It's okay.
It's okay. I'll probably make the same mistake.
I got it.
I did it for you.
Thank you.
Great. Okay, I'm gonna start from the top.
All right. Okay. So our second speaker is Angela Paul, and Angela Paul is a student in Dr. Valerie Gravel's lab. She started in the fall of 2018. Angela is originally from New York State, where she earned her bachelor's in environmental science from Wells College in 2018. And then before coming to McGill, she had the opportunity to work in a plant science laboratory at University College Cork in Cork, Ireland, where she discovered her passion for plant science. And then during her time at McGill, Angela has also volunteered as the Quebec representative for the Canadian Society of Horticultural Science, where she works. She worked alongside other reps throughout Canada to promote the study of horticultural sciences within Canada, Canadian universities, and other communities. Going forward, Angela hopes to continue working within the realm of horticultural science and furthering her work in fertilizer management and soil health. And so Angela's presentation today is analyzing the effects of nitrogen on flower bud induction and winter dormancy in day neutral strawberry. Go ahead, Angela.
All right, thank you for the introduction. So good morning, everyone. Today I'll be presenting my master's final thesis, like Valeria said, on analyzing the effects of nitrogen on flower bud induction and winter dormancy in day neutral strawberry. So for my project, I looked at cultivated strawberry known as Fragaria X ananassa, which is a hybrid of two wild type varieties, F. virginiana and F. chiloensis, and makes up the majority of all marketed strawberry in the world today. So strawberry is an increasingly economically valuable crop within the world, seeing about a 4% increase in strawberry production annually. The introduction of day-neutral strawberry in the early 1990s also helped influence this increase in yield, as these varieties of strawberry allowed for a longer harvest and higher overall yields. And you can see this in the graph below too, with around 1991-92, when these varieties of plants were first marketed, and you can see the gradual increase in growth every year. However, day-neutral strawberry research outside of where they originally developed in California as well as Florida is very limited. So taking a closer look at strawberry production within Canada, Quebec contributes most of all the strawberry production within the country, about 48%. However, strawberry is an increasingly valuable crop within the country. It's seeing about 18% increase in farm gate value since 2015. Although strawberry is increasing in value, however, the production of strawberry in Canada is nowhere close to meeting the national demand of strawberry within the country. In fact, approximately 95% of all marketed strawberry within the country is imported from elsewhere, mostly the United States and Mexico. And to make matters worse, this import-export gap has been widening rather than starting to close. So the question is, how can we start to narrow this gap? And one of the proposed solutions is to further study of day-neutral cultivars within Canada can address some of these yield shortages. So what do I mean by day-neutral strawberry? So currently there are 3 varieties of strawberry on the market. The first are short day strawberry, which are the most common cultivars of strawberry grown worldwide. They tend to yield best in under 12 hours of daylight, hence the name short day, and will produce berries within a 4-week window, starting in mid-June and extending to early July. After this initial yield, they will remain in a vegetative state where they will only grow leaves and stolons for the rest of the season. And then they will start to internally grow flower buds throughout this time into winter dormancy. The second variety of strawberry are everbearing varieties. They are less common because they produce smaller fruit and more runners, so they're less economically valuable. However, they yield best in longer than 12 hours of daylight, which is why they're sometimes also called long day strawberry. And they produce berries twice during a season with a vegetative state in between these harvests. They also produce flower buds during this vegetative state, and then again after the second harvest during winter dormancy. So now to talk about the cultivar of interest, day-neutral. These cultivars were developed by crossing an everbearing variety along with a wild-type Ephraigeniana variety. And the reason these cultivars are so important is that they are able to produce fruit irrespective of photoperiod. This means that no matter the daylight's length, they are able to continually produce fruit, and they can produce fruit for up to 20 weeks between early June until mid-October when temperatures start to decrease. This, as you can imagine, significantly increases their yield potential. And during this time, flower bud induction is continuously happening through the crown. There is no period of ceasing of flower bud induction. Now to just talk briefly about strawberry physiology. The strawberry plant can be broken down into 5 main parts: the pedicel or leaf stalk, the inflorescence or flower stalk where the flower buds stem from, runners or stolons, which will then branch to produce runner tips, which when in contact with soil or soilless substrate will produce daughter plants that are genetically identical to the plant. And then the central part of the plant is the crown, which is where all these new shoots originate. This is the crown. And just taking a closer in-depth look of the crown, you can see here inside there is the apical meristem. And here, this is where differentiation occurs into either one of these different shoots, either into leaves, runners, or inflorescences with flower buds. So with this, it's been shown that there is a significant correlation between the differentiation of apical meristems and environmental conditions. One of these being nitrogen availability, which can significantly affect whether or not more flower buds or more leaves or stolons are produced. So I also want to touch on winter dormancy in strawberry since I considered it in my project. So winter dormancy is a naturally occurring process in strawberry. It occurs once temperatures start to decrease. So as winter comes, as right now, strawberry will start to decrease the amount of stolons and leaves produced, and then will completely halt growth once night temperatures fall below 4 degrees Celsius, except for flower bud induction within the crown, which will produce flower buds for next year's harvest. So it's been shown in the literature that nitrogen treatments given pre-dormancy, before this dormant state begins to occur, can significantly affect how many flower buds are produced within the crown, as well as the nitrogen concentration in the crown can also influence the survivability of plants through winter. So now to take a look at day neutral strawberry in Quebec. As I mentioned before, day neutral strawberry were first bred in California by the University of California in 1991. But because these cultivars were originally produced there, they grow most optimally in the climates seen in California, as well as Florida, in a warm, fairly dry climate. So since they were first produced on the market, trials have been growing day neutral strawberry outside of these zones in more northeastern climates, such as in Minnesota and Ontario. However, in these trials, farmers saw that there was a significant drop-off of yield starting in about July and extending into August compared to what it should have been from trials done in California. So they weren't entirely sure what was causing this decrease in yield, but it's believed it has something to do with environmental factors, some of which either being the temperature, the humidity, and of course the fertilizer practices. So because Quebec has a similar climate compared to Ontario and Minnesota, it's assumed that Quebec farmers that are using day neutral strawberry are also going to see these decreases in yield. So the question is, how can we optimize yield in day neutral strawberry to increase Quebec production and allow farmers to confidently select day neutral strawberry cultivars? So for my project, I wanted to particularly look at nitrogen and its effect on day neutral strawberry. So the two main things that I wanted to cover were the nitrogen concentration as well as the nitrogen source. So first, looking at nitrogen concentration, I found that the nitrogen concentration significantly affects the parts of the strawberry plant that will grow. So with too little nitrogen, you're going to risk lowering your yield potential. However, the same effect can happen with too much nitrogen, as the more you increase nitrogen, you're going to see more growth of only stolons and leaves compared to flower buds, which is going to also decrease your yield. As you can see, this graph here too, which compared yield and nitrogen treatments to the plant, you can see there's a positive correlation up to a certain point when you reach the maximum optimal yield around 160 milligrams per plant, and then you start to see a yield drop off as nitrogen begins to increase. So for this project, I wanted to find the nitrogen concentration balance that is most optimal for increasing flower bud induction within day neutral strawberry. Secondly, I also wanted to consider the sources of nitrogen because I found that that can also have a significant impact on how flower buds are produced. So I looked at 3 different sources of nitrogen. I looked at nitrate, ammonium, and urea, and I isolated each of these sources and tested them individually on day neutral strawberry transplants. From the literature, I found that ammonia tends to produce more stolons than nitrate, and then urea produces the greatest number of flower buds. However, this was in short day strawberry, not day neutral, so I'd like to see if this would also correlate to day neutral strawberries. So before going into my objectives and hypotheses, I just want to also briefly touch on strawberry transplanting since I used transplants for both of my experiments. So just to cover, strawberry transplants are the way that most Quebec farms grow their strawberry through annual transplanting. And they use 3 methods, bare roots, tray plants, and fridge plants. And for my experiment, I mostly use fridge plants, which you can see in this transplant cycle. It starts with strawberry runner tips from the mother plant, which are harvested in about August by cutting these runners off of the mother plant. They are then grown within the greenhouse to produce roots, and they're given pre-dormancy nitrogen treatment until the temperatures start to cool in about October. Once temperatures start to get very cold and the plants stop growing, they are put into cold storage to initiate dormancy, where they are kept until spring when the plants are removed, and then they are put into the field for production. And then once these plants mature and start producing more runners, the cycle repeats itself. But I wanted to cover this because for both my experiments, I used the transplant stage for both production and pre-dormancy. So now to cover my objectives. So I broke down my project into two main experiments. The first was transplant production. So for this experiment, I wanted to determine the nitrogen source and the nitrogen concentration that resulted in the greatest induction of flower buds in day-neutral transplants. The second experiment I wanted to look at was the pre-dormancy, starting in about July and August, and going up until October when plants were put into cold storage, and again determining the nitrogen treatment that resulted in the greatest number of flower buds produced within the crown. And within this experiment, I also wanted to consider the role of potassium, as it has a key role in nitrogen and potassium assays. So my hypotheses for these objectives were that, one, we can significantly increase strawberry yield in Quebec by optimizing nitrogen practices in day-neutral strawberry. Secondly, I believe that a high concentration of urea of about 150 milligrams per liter per plant will result in the greatest increase in flower bud induction within day-neutral strawberry. Similarly, I believe in my second experiment that a similar treatment of 150 milligrams per liter of urea would have the same results and would produce more flower buds within the plant leading up to dormancy and then through dormancy in the winter. So going into my first experimental design, this was in production. So for this experiment, I looked at 9 treatment groups. The first, or the way I broke it down, was into the 3 sources, nitrate, ammonia, and urea. And then within each of these sources, I compared 3 different concentrations at 50, 100, and 150 milligrams per liter per plant. So setting this up, I took 162 day-neutral transplants in the cultivar Albion, which is a day-neutral strawberry that is more suitable for colder climates for Quebec, and grew them in the greenhouse in their greenhouse conditions with 12 hours of lights and about 25% temperature, so this wasn't variable. So I assigned 18 plants to each of the treatment groups and then gave nitrogen and calcium treatments bi-weekly, and I gave 50 milliliters of each treatment to each plant at each of these watering times. Along with the nitrogen, I also had to balance the potassium since I used potassium nitrate for one of my treatment groups. For the ammonium and urea, I also used potassium sulfate in order to keep the potassium the same. Then with all other nutrients, I used drip irrigation by just applying 3 pulses of fertilizer at a 180 dilution daily. And for each of these treatments, I also tried to monitor pH and EC, and keeping within an ideal pH range of 6.5 to 5.5, and then ideal EC of under 1.8 decisieverts. So for this experiment, I wanted to consider 3 main parameters, the first being the relative growth rate. So this calculated the rate of growth between treatments to see if one treatment increases the maturation rate of the plant, allowing for earlier yields. The second thing I wanted to look at was the weekly phenology by counting weekly counts of leaves, flower stalks, flower buds, and runners to see if any treatment had an effect on more flower bud and inflorescence production compared to leaves or runners. And then lastly, I also looked at the photosynthesis rates using a Lycor Ly680 photosynthesis system. And I measured the rate of CO2 gas exchange in the leaves of the strawberry transplants, hoping that one of the treatment plants would have a more rapid metabolic rate, which could also improve plant growth, plant yield, and the amount of flower buds produced. So my results. So first, looking at the relative growth rates, I compared this beyond all treatments. I calculated the relative growth rate using this formula here, where I compared the dry weight of a plant after I had finished data collection and applied all of the treatment after the period of my experiment. And then I subtracted it from the initial weight before I started applying treatments, which I collected at the start of my experiment. And then I divided that over the initial time of 0 days and then final time of 34 days. So calculating that, I calculated out the relative growth rate and then compared it using two-way ANOVA. However, from my results, I found that there was no significance of variance between any of the treatment groups in either the source or concentration. So the p-values here you can see are non-significant, which suggests that no one treatment allowed for a greater rate. Next, I looked at phenology, and in particular, I wanted to look at the amount of flower buds produced. So for this, I similarly counted all of the flower buds and other plant parts through every week. I compared the data using two-way ANOVA, and I did not find a significant correlation between any treatment except for the greatest concentration of urea at 150 milligrams per liter, and then the highest concentration of ammonia at 150 milligrams per liter. So for this, what my data suggests is that if you use 150 milligrams per liter of urea instead of 150 milligrams per liter of ammonia, you will have a greater amount of flower buds produced. But among all other treatments, this was not significant. And then lastly, I looked at the photosynthesis rate. And for this data, I did find that there was a significant amount of CO2 gas exchange in all of the urea treatments compared to both the nitrate and ammonia. So this suggests that the photosynthetic rates of urea is a lot higher than that of the other two sources of nitrogen, which could allow for greater yield and greater flower bud induction. However, from this experiment, I did have a few setbacks. One was that I did have a lot of calcium deficiency. However, I noticed this among all the treatments. There was no bias between one treatment. And as you can see from this lovely picture here, my poor strawberry leaves are all shriveled up because there was a bit of calcium deficiency. And this was mostly because I had to use a calcium substitute because I could not use calcium that had nitrogen in it, as it would conflict with my treatments in isolating the different nitrogen sources. So I don't know if this deficiency had any effect on the collected data, but because I did see significance in between treatments, I think it might have been okay. The only other issue that I did have is I saw that there was pH variation among the nitrogen sources. The pH was significantly lower in ammonia and urea treatments, despite me trying to combat that with increasing the pH. However, I still saw significance within the urea treatments, so it's likely had no significant effect on growth. So from this experiment, I concluded that the phenology data shows that urea at 150 milligrams per liter produces significantly more flower buds compared to 150 milligrams of ammonia. The relative growth rate analysis determined that there was no significant variance in the rate of growth between treatments. However, the CO2 gas exchange rate from the photosynthesis assay found that there was significance in all the urea treatments, which had a higher rate of photosynthesis compared to nitrate and ammonia. So the hypothesis is supported in the case of 150 milligrams per liter of urea compared to ammonia. But other than that, it's not supported. So now moving on to my second experiment, which was looking at the pre-dormancy trial. So for this, I broke down this experiment into two trials. The first trial, again, compared the different nitrogen sources and concentrations. And I followed almost the exact same treatments as my first experiment, looking at nitrate, ammonium, and urea. But then I also considered a control group using a nitrogen treatment that's used by farms within Quebec for day neutral strawberry, just to see if there was any correlation between that as well. So for this, I compared the same concentrations, and then the control stayed at 120 milligrams per liter of nitrogen per plant. So for this experiment, I grew 513 runner tips within the horticulture center here at McGill University. I assigned 51 plants to each of the treatment groups, and transplants were grown for 6 weeks in the horticulture center, with data collection beginning in September 4th and going until about October 18th. So similarly, nitrogen treatments were delivered at 40 milliliters per plant, 3 times weekly. And then potassium was also balanced for this experiment as well to keep that consistent. And then all other nutrients were also delivered by hand when needed each week. And And then the control treatment followed the same recipe used by the Onsmipua farm, which I'll talk more about in my next slide. And then after the 6-week period, after data collection, I moved the transplants into cold storage to mimic the practices in the farms to initiate winter dormancy, where I continued to take data throughout the winter. So then the second part of this experiment, I wanted to compare the nitrogen and potassium ratios because I felt that potassium could also have a significant effect on flower bud induction. So for this experiment, I compared 3 different ratios of nitrogen to potassium. I looked at a 1-to-1 ratio, a 1-to-2, and then a 1-to-4 ratio. And then nitrogen, similar to the control in my first part of this experiment, was at 120 milligrams per liter per plant. So for this experiment, I worked in collaboration with the Onsmipua farm in Ile-d'Orléans, Quebec, which you can see in the map below. It's close to Quebec City on this island here. And within this experiment, they let me have 3,000 transplants with 1,000 transplants assigned to each of the treatment groups. And for each of these treatments, the nitrogen to potassium ratios were delivered by sprinkler system by the farm. And then experimental data collection coincided alongside trial 1 with data collection occurring on October 5th and then going until October 19th to keep that consistent as well. And then transplants were also moved to cold storage by the Pulua farm staff starting on October 23rd in 2019. And then I continually took data and collected plants throughout the winter. So for this experiment, I considered 4 different parameters. Again, I considered weekly phenology, counting the different leaves, flower stalks, flower buds, and runners. I also wanted to look at the dissection of crowns by opening up the crowns and looking to see the flower bud induction happening within the crown and count the amount of flower buds that I found. I also wanted to compare biomass and the overall dry weights between the treatments to see if there's any difference there in the amount of plant growth leading up to dormancy. And then I also wanted to compare the crown sugar analysis by looking at glucose, fructose, and sucrose levels within the crown, as this could determine how well the plant survives through winter and how well nutrients are being utilized within the plant, allowing for more flower bud production. So unfortunately, with my trial 1, looking at the different nitrogen sources and concentrations, I found that I did not have any significant data between any of the crown dissection, phenology, biomass on trial 1. So this suggests that the source and concentration of nitrogen likely does not play a significant role in flower bud induction in pre-dormancy treatments for day-neutral strawberry. But moving forward, I did find significance between the trial 2, looking at the different nitrogen to potassium ratios. So first, I'm looking at the crown dissections in the different ratios, and this was compared using 2-way ANOVA. So for this, I found that there was a significance between the 1-to-2 and decay ratio compared to the 1-to-1 and 1-to-4 in both January and February with the amount of flower buds found within the crown. So with this being significant, this suggests that supplying 2 times this amount of potassium to day-neutral strawberry transplants can likely increase the amount of flower buds produced leading up to dormancy and during dormancy. Then looking at phenology, I also compared this using ANOVA. And I did find, again, a significance between the 1-to-2 ratio of N decay compared to the 1-to-1 and the 1-to-4. So for this, in all the weeks that I tested leading up to the dormancy in cold storage, there was a significant amount of flower buds seen and observed in the 1-to-2 ratio compared to the other 2 ratios, again, affirming that 2 times the amount of potassium will likely increase the amount of flower buds produced during pre-dormancy treatments for day-neutral strawberry. And then looking at biomass, I also found there was significance, again, in the 1-to-2 ratio. with the most amount of, or the highest weight of plant growth in the 1:2 ratio compared to the 1:1 and to 1:4. And then you can see as well, and I took some comparison photos of the different transplants from each of the treatments. You can see that the 1:4 are a lot smaller with less root development. And then 1:1 is about standard. And then 1:2, the roots may look a bit smaller, but there's a lot more leaf growth and stolon growth and also inflorescence growth. It's a bit hard to tell, but there was a lot more flower stalks within this treatment group compared to the 1:1 and 1:4. And then with the sugar crown analysis, I wanted to measure the total free glucose, fructose, and sucrose count within all of the crowns on both treatments. And to do this, I would prepare crown material by grinding it and then boiling it in 80% ethanol to separate the sugars from the plant material. Then using a glucose hexokinase assay, the glucose within the crowns was converted to gluconate, which is then readable at 340 nanometers in a microplate reader, allowing me to analyze the rough amount of glucose within the crown. And then sucrose and fructose were also analyzed by first breaking them down into glucose using different pathways, as you can see here. And then once they were broken into glucose, that was measured as well using a microplate reader. However, because of COVID, these results have been delayed. So unfortunately, I do not have the results today to present to you, but they are on their way to being finished. So hopefully we will see some significance there and perhaps even see significance in the first trial and see maybe what's going on with why there's no significance in the other parameters. So from this experiment, I concluded that trial 2 phenology data showed that a 1:2 ratio of NTK produced significantly more flower buds than 1:1 and 1:4 at both the phenology state and then also within the crown by doing crown dissection. Biomass also showed a similar result with the 1:2 ratio having the greatest amount of biomass compared to the other two ratios as well. And then again, trial 1 did not have any significance, which suggests that perhaps that the nitrogen source and concentration is not significant on how flower buds are induced at the pre-dormancy stage for day neutral. So the hypothesis was rejected for this experiment that 150 milligrams per liter of urea would produce the most flower buds. So that would be rejected. So then my final conclusion to go over again, from experiment 1, 150 milligrams per liter of urea produced significantly more flower buds than 150 milligrams per liter of ammonia. And then all urea treatments had significantly greater rate of photosynthesis. And then the relative growth rate did not have any significance between treatments. So that's that. And then again, in experiment 2, the pre-dormancy treatment saw no link between nitrogen treatment and flower bud induction. And there was more flower buds were induced in the 1:2 ratio compared to the 1:1 and then the 1:4. So how does this contribute to your research? So from my experiment 1, it can be suggested that the isolation of urea fertilizer, that urea at higher concentrations will tend to have a significant effect on flower bud production and photosynthesis rate in day neutral strawberry. Therefore, farmers can tend to use more urea in their treatments, and that could perhaps increase the amount of yield that they see with their day neutral strawberries. Then a 1:2 ratio of N to K is also found to be most optimal during pre-dormancy. So using that instead of a 1:1 ratio would be more optimal for pre-dormancy treatments. And then going forward with this research, I believe this experiment could be repeated, perhaps using other day neutral strawberry cultivars. For my experiment, I only focused on Albion because it is the day neutral strawberry cultivar that's most used within Quebec since it has greater cold immunity. But we could also test with other cultivars to see if there's any difference because there are other cultivars being used within Quebec right now. And then also further parameters can also be tested. We can look more into the microbial communities on nitrogen uptake and see the interactions with that. as well as maybe look at other environmental factors and how they affect the flower bud induction, including temperature, since that tends to play a larger role in induction of flower buds. And with that, I would like to conclude, and I would like to thank my supervisor, Dr. Valerie Gravel, for all her help and support through these 2.5 years. I'd like to thank Dr. Phillips again, my committee member. I'd like to thank Dr. Jacqueline Bede for letting me use her microplate reader in her lab for this last sugar analysis. I'd like to thank the Onsimipuwa Farm for letting me use their facilities and giving me lots of plants to analyze for them. I'd like to thank NSERC for funding and my lab members for all their help and support throughout the year. And then also the plant science department. Thank you all so much for this and helping me through this journey. So thank you. And that's it. And if you have any questions, I'd be more than happy to answer them.
Okay, thank you, Angela. Again, if you have any questions, go ahead and unmute yourselves, write it in the chat, or write it on the YouTube channel. So, do you know if there are strawberries how are they bred? From the breeding point of view, do you get somoclonal variants like you do, say, in grapes?
I'm not entirely sure because mostly what I use would be like the transplants, which are supposed to be like genetically identical to the mother plants, but I'm not entirely sure as far as the breeding thing goes, if there is variation, because that would be something to look at too. Maybe look at the different genotypes and see if maybe there is variation between the transplants, and that could have an effect as well.
I know strawberries have a very complex genome biology. They're octoploids.
Mm-hmm. Yeah, for sure.
Yeah.
I have a question. Oh, sorry, there's one in the chat. Never mind.
Okay, I think the one in the chat came, but yeah, hold your question.
Oh, I didn't see that there's actually a chat too. Okay.
Yeah, there's two questions on the. I'll ask and.
Okay, yeah, I can't see them, but.
Did you provide any calcium supplement on your second experiment to avoid deficiency? Is the first one.
For that experiment, yes, I used a foliar calcium spray, and it seemed to work a lot better at that stage compared to using, because for my first experiment, I just used like calcium solution instead of a foliar, we call it application, but the foliar application seemed to work better, and I didn't see any deficiency with that, which was good.
If you had the chance to use the microplate reader, what would you expect your results to be?
I would hope that I would see there would be differences in the amount of glucose and other sugars within the crown, especially within the urea treatment. I feel like that would likely have more sugar production, and also just to see for survivability through winter as well. But I cannot say entirely from the research. There hasn't been much looking at the different sources of nitrogen and then sugar content in the crown. So I can't say for sure if there would be likely one treatment that would be more likely to have more sugar than the others. But yeah, so it'd be interesting to see.
Okay. Did we have one via the microphone still?
Yeah, I was just wondering, so your focus was on Canadian and Quebec strawberries, but is this research applicable to global strawberry production?
It could be. So I mainly wanted to focus because obviously there's a big yield gap between imports and exports of strawberry within Canada, in Quebec in particular. So that's where I mostly focus. But yeah, I mean, looking at producing dangerous strawberry outside of just California and Florida is very important because there could be significant yield increases if we could. But right now, with the way things are, they're just not performing as optimally. But there are also breeding programs going on right now, looking at finding the neutral strawberry that are more susceptible to colder climates and the climates in Quebec. So looking at that too, to increase flower bud would be good.
Excellent, thank you.
Any other questions? Well, if we don't have any other questions, let's thank our presenters. And please don't forget to fill in your survey on my courses. And we'll see you next week. Next week we have, let me see, Anis Fatmawati from the Sing Lab and Luke Willett from the Sharon Lab. So see you next week. Thank you.
Good job, Angela.
Thank you.